Can you
Good
see my
afternoon,
screen?
participants. A very warm welcome to Dr. Manideepa Banerjee. May I introduce our speaker, Dr. Manideepa Banerjee, who is an associate professor of Kusuma School of Biological Sciences, IIT Delhi. A brief bio on Dr. Manideepa Banerjee. She received her bachelor degree in chemistry and master's in biochemistry from Calcutta University. She received her PhD from University of California, San Diego, where she studied the molecular mechanism of bacterial pathogen entry into host cells using biochemical and biophysical methods. She carried out her postdoctoral research at the Scripps Research Institute, California, where she received extensive training on X-ray crystallography, electron microscopy, and cryo-electron microscopy, and 3D reconstruction of biological micromolecules. Dr. Banerjee returned to India as an assistant professor in IIT Delhi and a DBT Ramalinga Fellow in 2010. Since then, she has set up a facility for cryo-electron microscopy at IIT Delhi, which is being utilized extensively by academic institutes all over India. The research interests of her laboratory are in virus force interaction as well as structural analysis of biomolecules using electron microscopy. I truly welcome you, ma'am, and I would like you to proceed with your session. Thank you.
Thank you, Risha, for that very nice welcome. Are my slides visible on screen? Hello?
Hello. Yes, ma'am.
My slides are visible on screen?
Not yet, ma'am.
Okay, because I've shared them.
Slides are visible, sir? Yes, ma'am. Slides are visible.
They're visible?
Yes, ma'am.
Okay, shall I start?
Yes, sure, ma'am.
Okay, all right. So good afternoon to all the participants in the workshop. It's a great pleasure to be here, although virtually. I have been asked to talk about electron microscopy and cryo-electron microscopy, the recent advances in applications. So that is what I will do. And if there are any questions, I assume that people can raise hands virtually and I will try to address them. So when it comes to microscopes, I think this figure that I'm showing is familiar to all, the principles of image formation. So it's essentially an object can be magnified through a microscope so that we can visualize the details of the object in an image which is magnified by the microscope. So the type of microscope that one uses, or let's say the resolving power of microscopes, or what illumination source needs to be used for visualization, depends on what kind of object we're trying to see. There are objects which we can just see by naked eye. There are objects for which we need a light microscope. And then there are very small objects like, for example, a virus particle or a protein complex for which something better is required so that we can see material that is so small. So to address these issues, to be able to visualize particles that are very, very small, the electron microscope was developed. The first electron microscope was designed and built by Ernest Ruska, who you see here in the photograph, in 1931, for which he received the Nobel Prize. There were some major discoveries in physics which paved the way towards development of the first electron microscope. Some of these discoveries are that accelerated electrons behave like light in vacuum. They can travel in straight lines and they have wave-like properties as well as particle-like properties. Electromagnetic fields can be used to bend and focus electrons, which is very important when you are trying to use them as a source of illumination. And what is very important is that the wavelength of electrons are 100,000x shorter than that of visible light, which means that they can be used to illuminate very, very small samples, which visible light cannot do. So based on all of these discoveries, Ernest Ruska developed the first microscope, and this is what it looks like. Um, just to show you how far we have come, the resolution of the first electron microscope was 100 nanometer. These days, some modern light microscopes have resolutions of 200 nanometer. So obviously, the resolution of electron microscopes have increased quite a bit. And these are some of the advances that we're going to be talking about. However, even though there have been significant improvements, the overall schematic of a transmission electron microscope remains more or less the same. So you have an electron gun. This is the area that generates electrons, which are used as illumination source. Now, after this, there are a series of condenser lenses, which convert the electron beam from a diverging beam into a parallel beam, after which the electrons they fall on an object, they're transmitted. Then the objective lens produces the first initial magnification. This is followed by intermediate and projector lenses, which further magnify the image, so we are able to see things in great detail. This is what a modern day transmission electron microscope looks like, and the resolution is much, much better than it was when it was initially developed. Now, a scanning electron microscope is a little bit different in plan, in the sense that there is an electron gun, there is production of electrons as a source of illumination. However, essentially what we do here is that we get an idea of what the surface of the sample looks like. And the electrons, after hitting the surface of the sample, generate secondary electrons, which are detected by detector. So again, the scanning electron microscope, which was first described by Max Knoll, has gone through many iterations, and now it is a much better, and the resolving power is much better. So let us take the components of electron microscopes once at a time. One of the very important components is the source. Source is the material that generates electrons. And there are different types of sources in an electron microscope. And these in different electron microscopes, which decides basically the power of the microscope, what is the magnification, energy distribution, etc. So one of the very important sources is a tungsten filament. This is what a tungsten filament looks like. The filament is heated to a very high temperature. As a result, there is thermionic emission of electrons, and the electrons are accelerated by the electric field between the anode and the filament. Tungsten filament generally produces a magnification of about 50,000 x, and the energy distribution is about 2.5 electron volt. This tells us that the energy beam is parallel, but it can be better. The next type of source is lanthanum hexaboride crystal. This is what it looks like. This again undergoes thermionic emission to generate electrons, and the electrons are produced from the crystal vertex right here. Lab 6 has a lower work function compared to tungsten, so you need a little bit lower temperature, and it has a better brightness and lifespan. The magnification also is significantly better. Magnification can be up to 100,000 x, and the energy distribution is also better. Now, for cryo-electron microscopy and application to biological samples, what I'm going to talk about today, later on, the source which is primarily used is called a field emission gun, or an FEG source. So this is essentially a single crystal tungsten tip, and it is sharpened to between 10 to 25 nanometer. And this is coated with zirconium dioxide, which further lowers the work function. So the electrons are thermally emitted, and then they're extracted by very strong potential gradient and accelerated through various voltages depending on depending on the type of microscope. The beam generated is much more bright than that done by tungsten, very, very coherent. You can see that energy distribution is just about 1 electron volt, and this generates magnification higher than 100,000x. Now, apart from the Schottky type FEG or heated FEG, there is also a cold FEG where no heating is required, and this has even better brightness and more coherence, and it has a little bit more intense maintenance. Lenses, again, are an integral part of any microscope, similar for electron microscopy. However, here the lenses are electromagnetic lenses, and the power of the lenses can be altered by varying current in the coils. Like other lenses, optical lenses that we typically use in microscopes, electron microscope lenses also have similar types of aberrations, like spherical aberration, chromatic aberration, or astigmatism. So what is spherical aberration? In this case, the rays passing through the lens periphery become more strongly refracted than paraxial rays, like these ones. And the correction of aberration depends on the manufacture of the lens or design. In fact, we will see that the spherical aberration of value of a cryo-electron microscope is a very important parameter. A chromatic aberration is when longer wavelength rays get focused more strongly, and this results in a halo effect. So there is a colored halo around images. This is an example of chromatic aberration, and this can be fixed by a more stable accelerating voltage. Astigmatism is caused by asymmetric magnetic field in the lenses when a sphere becomes an ellipse. So this is an example of astigmatism, and this can also be addressed by altering changes in stigmata points. Now let's come to what is the principle of image formation by an electron beam. So when incident electrons fall upon an object, there may be elastic collision of electrons with atoms, or there could be inelastic collisions. In case of elastic collisions, the electrons are scattered with no change in kinetic energy, like this one that you see here. And these are the electrons which will eventually lead to the formation of an image. On the other hand, inelastic collision leads to alteration in kinetic energy. This can change many things. Primarily, this contributes to noise. Now, when we talk about biological samples, because that is what we're going to focus on today, one thing to keep in mind is that biological samples consist of light atoms with less number of electrons, like carbon, hydrogen, oxygen, nitrogen. So these are what are called weak phase objects, meaning they don't absorb the source of illumination. They will just cause a slight alteration in the directionality. So what is this generally called is these will cause a phase change, not too much of an amplitude change, which results in very low contrast. So if you see here in this image, these are the biological samples, but it's very, very hard to see the samples among the background. So there is really a lot of noise. But recent advances has resulted in getting images like this, where at least the biological samples can be clearly visualized. And later on in the talk, we will discuss how the generation of contrast in images can be altered or can be improved significantly using this process. So, as we were discussing, when essentially a beam is absorbed by an object, this gives rise to what is called amplitude contrast. As a result, you have this pattern of darkness and light, and this is what is contrast in your image. The problem is biological samples, as we discussed, because they're weak phase objects, they do not absorb beam. Rather, they cause a little bit of a phase shift. So as you can see, the directionality of the transmitted electrons change. So the emergent beam that is coming out is undergoing either constructive or destructive interference with the beam that is parallel, and these phase variations generate whatever contrast that we see. But there are ways in which phase variations may be converted to amplitude variations. So in optical microscopy, This is done by introduction of something called a phase plate. This is introduced in the back focal plane of objective lens right here, and this shifts the phase of scattered electrons by 90 degrees. Now, if you can put a wave, an outcoming wave in a complex numbered plane, you will see that if the phase is shifted by 90 degrees, it essentially, the phase alteration is converted to an amplitude alteration. So this gives rise to more contrast. So as you can see, this image is without a phase plate, this image is with a phase plate. So similar kind of situation can be carried out in a transmission electron microscope, where a phase plate can be introduced at the back focal length of objective lens to create this alteration in contrast, which provides more detail about the particles that one is working with. Now, electron microscope in general is phase contrast microscopy. So there are several methods by which contrast can be generated or is generally generated in electron microscopes. One of them is spherical aberration. So spherical aberration we discussed as a property of the objective lens of the microscope, and this essentially generates a little bit of a phase contrast, which can be exploited to visualize objects at a higher with a bit of a contrast so that more details are visible. Another way to improve the contrast is to remove the inelastically scattered electrons which generate noise in the image. And this can be done by playing with the apertures in the microscope. The third option is defocus. So when we typically take an image, we take the images in focus. But defocus can be used to improve contrast. So let me go through these three methods a little bit in detail. In case of spherical aberration, diffracted rays with higher angle of incidence converge before the focal point, not at the focal point. What this does is that it generates a phase shift. In case of noisy images, the apertures can be manipulated to remove the noise causing inelastically scattered electrons. So this also improves the quality of the image. And finally, defocus, which essentially means taking images a little bit of under focus. So again, there is a little bit of phase shift in the transmitted electron beam. And together, if you combine all of these three things, this generates a decent amount of contrast in images so that particles or biological samples, this is specifically for biological samples, this can be seen clearly. Now, another way to improve contrast and to see biological samples more clearly in electron microscopy is energy filtering. So this essentially means removal of inelastically scattered electrons by using a filter. And the filter can either be in the column of the microscope or it can be post column. So just to give you an example, this is an image of bacteriophages. These are viruses that infect bacteria. And these images, the top images, can without using an filter. And you can see that as an energy filter is being used, the images are much more clear. Similarly, here, the filaments can be visualized very nicely by introducing an energy filter. Now, another important advancement or improvement in recent years has been changes in the detection system. Detection system is whatever registers the information coming from the microscope and generates an image which can be or provides an image which can be visualized. And typically, the detection system has been CCD cameras, which are charge coupled device cameras, where incident electrons are converted to photons. So there's this multi-stage conversion. And then eventually this charge readout is converted to pixels, which are then available for us to see. Now, the latest advancement has been CMOS cameras, which directly convert electron energy without this multi-stage conversion. So this saves quite a bit of information or prevents them from the information from being lost. And this has led to the formation of the development of what are called direct detector cameras, which use CMOS technology. Plus, the images are taken in what is called a movie mode. So here, if you can imagine an electron beam hitting a very small sample, sometimes what happens is, or it what happens is, that the sample can move a little bit. There can be a little bit of movement. As a result, there can be quite a bit of blurriness that is created in images. Now, direct detector cameras take images in movie mode instead of in still mode, and then correction is carried out to ensure that this blurriness is removed. And now you can see that the images are much, much clearer in direct detector cameras. So this has also been a huge advancement in electron microscopy. Energy filtering as well as direct detector cameras really improve the quality of images and really generate images with high contrast. However, regardless of what are the different advanced methods that are being used to record images, one has to keep in mind that whatever image that we see is never a completely 100% faithful reflection of the image, because there are a lot of properties of the microscope, the environment, the sample, etc., which can potentially affect image formation. So I have listed some properties which can affect image formation. There could be other things. There could be significant aberrations in lenses. The source may not be coherent. There could be significant sample drift, probably because of some instability in the environment or in the microscope itself. There could be alterations in lens current. There could be some quantum noise as electrons hit the sample. There could be, in case of cryo-electron microscopy, as I will describe a little later, the quality of ice becomes very, very important. So all of these converge to a point where the image is not 100% reflecting the object. So these properties of the microscope or the environment, which lead to aberration in the image, is called a point spread function, or PSF. So essentially, if you take the object and combine it with its PSF, you get the image, which is not, as I was saying, a complete representation of the object, because all the imperfections in the system are transferred to the image. So this is kind of, reminiscent. I think you are all familiar with rangolis that are done in hands, and then mehendi that is done in hands, and rangolis that are done on surfaces. So essentially, your microscope is like a paintbrush, and if your microscope is a very fine paintbrush, then all the finer details can be captured. If it is not, then the details that we capture may not be as fine, and we may be missing details. So this is just a way of saying, what is the point spread function and how does it relate the object to the image. So this function called the contrast transfer function, we will come back to it at later in the talk. So this is a Fourier transform of the point spread function, and the images have to be corrected by this value in order to correct for imperfections that are there in the environment, or in the microscope environment. Now we come to usages. So usages of electron microscopes, as I think we know, these can be used for EELS, which is an electron energy loss spectrometry, which can be utilized to figure out elemental analysis, surface properties of materials, X-ray microanalysis using EDX, dual beam SEM, where it does high resolution SEM analysis. So these are all different types of usages that can be carried out. But today our focus is going to be on biological samples, or application of electron microscopy to life sciences. So this is what we will focus primarily, which is structural biology. I think you may be familiar with structural biology methods like X-ray crystallography or nuclear magnetic resonance, NMR. Cryo-electron microscopy, rather, is an alternative method of looking at 3D structures of biological macromolecules, which has become very, very popular recently. And we will discuss how that is done. In addition, there are topographic methods which can be utilized to look at relatively thicker samples, and hybrid methods which combines cryo-electron microscopy with either NMR or X-ray crystallography. Apart from this, in cell biology, cryo-electron microscopy or electron microscopy is used to look at cellular morphology, organelles at high resolution, tissue ultrastructure, as well as biomaterial. So let us go to applications of electron microscopy to structural biology. So when we say structural biology application, we generally are talking about 3D structures. Typically, images that we collect in electron microscopy are 2D, they are flat images. And this method is utilized, so cryo-electron microscopy and single particle reconstruction, or SPR, is used to reconstruct these flat images or 2D images into 3D density volumes. And when we're talking about, again, structural biology or small molecules, here you see images of a virus particle, which has been done in my laboratory. You can look at protein complexes or protein molecules. So you can imagine that we are looking at very, very thin samples because we are looking at individual protein molecules or individual protein complexes or individual virus particles. So the samples are very, very thin. And usually there is a significant amount of radiation damage in biological samples when due to electron energy. So here you can see how a sample degrades over time with increased amount of electron energy. Now, so this is where the importance of cryo-electron microscopy comes in. So what typically to look at objects, biological samples, and to increase contrast, people use a method called negative staining. So you see here in these images, there is negative staining done on the particles, which essentially means that you add some kind of a heavy metal which shadows the particles. And as a result, the particles are very clearly visible. But what negative staining also does is that it obscures all the finer details because you have a thick layer of heavy metal falling on your sample. So a lot of details which improve the resolution of the object is obscured. So when we say cryo-electron microscopy, essentially the particles are quick frozen, and we will discuss this in detail later. They're quick frozen in amorphous or vitreous ice. As a result, all the details are preserved. So the particles are preserved in what is called near native state. And this becomes very, very important in order to resolve finer details. So this method for using electron microscopy for determining 3D structures of macromolecules is called cryo-electron microscopy and image reconstruction. And this has become very, very important, a very important method in the recent years. This is because this method has significant advantages over other structure determination techniques like X-ray crystallography or NMR. This method requires significantly less material. For X-ray crystallography or NMR, one first has to concentrate biological samples to 5 milligram per ml or 10 milligram per ml. In case of X-ray crystallography, one first has to get crystals, which would diffract, and then data can be collected, and the structure can be resolved from the data collected. And this all takes a lot of time. However, in cryo-electron microscopy, there is very little material required, significantly less time is required, no need to crystallize biomolecules. Now, this part is very important. In using cryo-electron microscopy, it is possible to analyze samples which are very heterogeneous, which typically is a big problem in X-ray crystallography or NMR. Cryo-electron microscopy was initially used to analyze large complexes because it was very difficult to look at single protein molecules in cryo-electron microscopy. But I'll show you some examples as to how it has become possible in recent years. It is also now possible to achieve very high resolution using cryo-electron microscopy which again was not possible just about 10 years or so ago. So cryo-electron microscopy, as I discussed, essentially means that you are looking at frozen biological samples. And this method of freezing was standardized or developed by this scientist, Dubochet, who received the Nobel Prize in cryo-electron microscopy for developing the plunge freezing method. And in this method, essentially, the sample is frozen very, very quickly, very fast in liquid ethane, which is being cooled by liquid nitrogen, so as to generate amorphous ice, which does not hurt the sample, it does not harm the sample or alter any of the outer surface details, but all the details are preserved and the sample is visible in near native state. So as the sample is undergoing Brownian motion in a tube, let's say you purified your protein or you purified your virus, it is present in a tube. Now it is undergoing Brownian motion. As it is immediately frozen or very quickly frozen, the samples, individual particles are frozen in all different orientations. And having all different orientations of a biological macromolecule is very, very important in order to resolve the 3D structure. So let me first give you a very quick demonstration of how sample freezing works in electron microscopy. So essentially, you take something called a grid, which is a 3 millimeter diameter, a small spherical object. And this has to be handled using tweezers. And on the grid, sample is added. As I said, the volume of sample required or the concentration required is very less in cryo-electron microscopy. Maybe 3 microliter sample at a concentration of half a meg per ml is sufficient. Now, this is typically frozen. It can be manually frozen very, very quickly. It can also be frozen using various types of robotic devices. We have a robotic device called a Vitrobot in our laboratory, where at a certain humidity or certain conditions of temperature, the sample is very, very quickly plunged into liquid ethene, which is being surrounded and cooled by liquid nitrogen. So from this point on, the sample has to be kept in very low temperature. It has to be minus 180, because if the amorphous ice devitrifies and then reforms, then you get crystalline ice, and that will damage the sample, or the sample would not be imaged properly. Now, this grid is put in the microscope, in the cryo-electron microscope. It is being cooled continuously because the holder that holds the sample has a liquid nitrogen reward attached at the end. And as the sample is put in, now we start magnifying the grid and looking at what is there. So this here is an image of the entire grid. You see there are a lot of grid squares. This is at 50x magnification. Then at 500x, you start seeing that there are these little holes in the grid squares. And eventually, you would see that there are, we'll come to this, you will see that the sample typically freezes inside these holes. Okay, now here I should mention, and we discussed this a little bit earlier, that when samples are or electrons are heating the sample, this is generating heat. And this results in degradation of biological samples. So the images in cryo-electron microscopy are typically taken in what is called a low dose mode, meaning that you are using a very low dose of electrons, and focusing and alignment is done on a different side. So as a result, the signal to noise ratio becomes very, very low. So here now we are looking at one of these holes in the grid square, and you can start looking at the particles that are frozen. These are all frozen in amorphous ice. These are virus particles in near native state. So all the details of the particles are preserved. Now, when we get these images, the images are flat images or 2D images, and now they have to be converted to a 3D density volume for the purpose of resolving a structure. So let me just demonstrate this a little bit. So here you have a molecule. These are three different orientations. It's a protein complex. And as the electron beam hits any of these orientations, you get these flat images that you see at the bottom. Now, using these flat images, you have to reconstruct the entire particle. So how does one do it? This is a very, very iterative process, which consists of first picking particles. So you will take a lot of images from 20, 30,000 of each of particle image to some people have done 1 lakh images or 1.5 lakh images. Now, this is followed by CTF. Remember, we discussed CTF as being the Fourier transform of the point spread function. So the CTF has to be estimated and the images have to be corrected. This is followed by extraction of particles and creation of stacks of particles which are in the same orientation, which improves the signal to noise ratio significantly. This is followed by alignment, 2D alignment, classification, creation of an initial image from 2D aligned images, initial model from 2D aligned images, and 3D reconstruction and refinement, followed by analysis of the density. So let's go through these steps. The first step is particle picking. So each image, this one is called a micrograph, each image is called a micrograph, has a lot of these particles. So expect if your sample quality is good, there should be a significant number of particles. Now, these have to be picked either by a program or manually. And as you can see here, there are particles in different orientations. For example, this one is in a different orientation as compared to this one. This one is in a different orientation. Now, one has to make stacks. So for particles in the same orientation, you will sort them into different subsets, as you see. And this improves the signal to noise ratio because remember that these are biological samples. So there is a significant amount of noise in the background, and improvement of signal to noise ratio is very, very important. Now, what this analysis also does is that it reveals very important information about the biochemical variations that could be present in a data set. For example, you may have a protein complex, which is a combination of a dimer and a tetramer. Perhaps this kind of association or oligomerization is a very dynamic process. And as a result, it's not possible to separate dimers into tetramers, dimers from tetramers using biochemical methods. Now, this mixture can be very nicely sorted out in this 2D classification stage, where you can have different stacks of either dimers or tetramers in different orientations, and they could be appropriately combined to get two different 3D structures as opposed to one. Now, once this 2D classification is carried out, then the different classes, which are all in different orientations, have to be pulled together to make a 3D model. So I will not go into too much detail about how to generate a 3D model. I just want to mention that there are several methods for doing this. There's a method which is a random conical tilt, where random projections are oriented or they come around to form a surface. There is a common lines method, which essentially searches for intersection of any two projections, and this is typically done in Fourier space, so that the projections can be assigned certain angular values. Then there's icosaedral reconstruction, which is typically applied for viruses, because viruses have a lot of symmetry, and that symmetry means that essentially certain fields of view are equivalent to other fields of view. So these can help in the reconstruction process. And then helical reconstruction, which is utilized for helical proteins or viruses or fibers. So these are all different methods of generating 3D models. And this is then combined to have an initial model. So you can see here how different orientations or projections have come together to form a 3D model. Now this is the 3D model, and this can be further refined. Refinement is very important. So essentially, the initial model generated is back projected, and the projections are matched with the original images, and the alterations or the refinement in angular values is done so that the 3D reconstruction becomes better, the resolution improves, or the correctness of the model improves. So finally, when there is no obvious improvement in resolution or correctness, then that is the final refined model. Now, once this is done, then one has to assess resolution. Resolution is typically assessed through a process called Fourier shell correlation cutoff. So the data set that one has collected is randomly divided into two, and the Fourier terms of the two halves are compared in different resolution shells. And there are different ways of estimating resolution from FSC. So there can be 0.5 cutoff or 0.33 cutoff or the gold standard cutoff. So there are different ways of estimating resolution. But eventually the information has to make biological sense. So the structure that is resolved eventually has to make biological sense. So I will just show you first, this was one of the first structures that were solved using electron microscopy. This is an icosahedral virus called tomato bushy stunt virus, and the structure was solved by Tony Crowther in 1971. So essentially particles were selected from micrographs, and essentially icosahedral reconstruction methods were applied, meaning that one field of view is equivalent to several other fields of views. And if the sets of particles were given some angular values, and there was a 3D initial model generated, which was eventually refined to generate the virus surface. So from flat images, this method was able to generate a 3D density volume image of tomato bushy stunt virus. So there are just some examples. This is an adenovirus, which causes eye infections, and there are other kinds of disorders. This is a fairly large virus, so solving the structure using X-ray crystallography, which was done by Vijay Reddy, was a very, very difficult and time-consuming process. And cryo-electron microscopy did not require to have a crystal of the virus made first, or no crystal manipulation was necessary. And the structure was solved at a decent resolution. So you could see a lot of structural detail that is visible here. And as you can see in the density, the X-ray crystal structure could be fitted. So this was an important advancement in cryo-EM. Now, in recent years, and the recent advancements that I'm going to talk about, there has been what is called a resolution revolution in cryo-electron microscopy. In spite of many changes, the plunge freezing method and other improvements in microscopes, the resolution of structures, 3D structures solved by cryo-electron microscopy for a long time stayed in general at around 5 to 6 angstrom, maybe around 10, 11 angstrom resolution for a very long time. However, in the last few years, thanks to the development of direct detection methods that we discussed, collection of images in movie modes, energy filtering, introduction of phase plates, there has been this revolution in the quality of images that are generated from cryo-electron microscopy, and as a result, resolution has improved significantly. And this is just an image which illustrates that. So this is beta-galactosidase, a protein molecule, and the structure was solved in 2005 using cryo-electron microscopy at a resolution of 25 angstrom. So you can see that the overall shape of the molecule is visible, but not much structural detail. Now, in 2011, as a result of this improvement in resolution, the structure was resolved at 11 angstrom. And again, you can see that although the overall shape is similar, but now we are starting to see a lot of detail about the structure of beta-galactosidase. In 2013, the structure was solved at 6 angstrom, so there is again a lot more detail. Then later on, the structure has been resolved at 2.2 angstrom. So you can see that now you can see individual amino acid side chains, which makes this a very, very important technique, because if you get this level of detail, then one can use this method very conveniently, not only to try to understand biological macromolecules, but also for application-oriented studies, like trying to identify binding partners or for drug discovery purposes. Also, as I had mentioned earlier, in cryo-electron microscopy initially, or even just about a decade ago, the size of particles had to be significantly large so that they could actually be visualized by this method. But now, because of the recent advances, it is possible to see even small protein molecules. For example, this is human hemoglobin, and this can be visualized very well through the electron microscopy. And a nice structure was resolved, which was published in 2017. Now, there are still significant improvements that are going on. These are images taken using a faceplate called the Volta faceplate. And if you look at these images, you see that there's a lot of contrast, almost reminiscent of using negative staining, but here there's no negative staining. But the structural details of the particles are coming out very, very nicely. So as a result of these advances in 2017, 3 pioneers in the field of electron microscopy were awarded with the Nobel Prize in chemistry. These 3 pioneers were Jacques Dubochet, who put the cryo in cryo-electron microscopy, Joachim Frank, who developed 3D reconstruction methods, and Richard Henderson, who determined or developed the direct detection method. So now we come to, since because of these advancements, we are able to see very high detailed or very high resolution or detailed structures of biomolecules. Why do we need this? Why do we need quickly high resolution structures of biomolecules or at high resolution? So first of all, this improves our fundamental knowledge. We need to know the way biomolecules work in order to understand how our body functions. Then this fundamental knowledge can be applied for drug development or drug assessment purposes. The sort of therapeutics that we take, all kinds of medications that we take, these are all targeted towards some kind of biomolecule in the body. So if we understand biomolecules very, very clearly, then drug development and drug assessment processes become easier, or they are more accurate, if not easier. Since in cryo-electron microscopy, a very small amount of protein is required, and the structure can be solved in days or weeks instead of years. So this becomes a very, very important method for this kind of analysis that we're talking about. For example, when the Zika virus was identified, then the structure was solved very quickly at 3.7 angstrom resolution. And this really is a great help in trying to develop therapeutics or vaccines against the virus. Now, we are all aware of the current pandemic caused by SARS coronavirus 2, which is happening currently. And this is why we're having a virtual webinar instead of having a real seminar with real interactions with other human beings. So, so here also, the goal right now is to develop some kind of a therapeutic or some kind of a vaccine candidate very quickly against the virus that is causing the pandemic. And here, cryo-electron microscopy and 3D reconstruction has had an enormous contribution, because one of the primary antigenic determinants of the virus is the spike protein, which is present on the surface. And the efforts to develop drugs or therapeutics has been targeted towards the spike protein. So it was very, very important to know what the spike protein actually looks like, so that this information can be exploited for development of drugs or therapeutic molecules. So the structures of spike protein in various conformations in conjunction with receptors, etc., were resolved using cryo-electron microscopy and 3D reconstruction. And this has really helped virologists and scientists all over the world to try to target specific therapeutic or drug molecules towards these proteins. So this is again another view of the spike protein. So having said that, I will in the end come to a brief description of the work that we do in our laboratory, where we are using or applying cryo-electron microscopy and 3D reconstruction. If you remember, we talked about how cryo-electron microscopy is applicable towards heterogeneous samples. Now, there are samples where there could be some kinetic parameters which ensures that they're heterogeneous, and it's very hard to purify different homogeneous populations from this heterogeneous sample. So resolving structures using traditional methods like X-ray crystallography or NMR becomes very, very challenging. So we faced this challenge when we were trying to identify the process or understand the process of virus disassembly. So as you know, viruses, when they try to infect a host cell, first they bind to some kind of a host cell protein or a receptor. Then the virus comes inside the cell, and it has to disrupt the cell membrane, or in this case, the endosomal membrane, in order to be released in the cell cytoplasm. Now, typically viruses, or in this case, we're talking about non-enveloped viruses. These don't have any lipid. The outer shell is an icosahedral protein shell. So these are very stable. But once the shell is inside the virus, it has to, inside the cell, it has to fall apart so that the genome can be released. So this process is called disassembly. And my laboratory has been trying to understand disassembly of icosahedral viruses for a very long time. So this is the step that we are interested in, the disassembly and genome release. Now, when you think about icosahedral viruses, these are very, very symmetric and very stable. And icosahedron, as we know, has many symmetry axes. So there are 12 5-fold axes of symmetry, 20 3-fold axis of symmetry, and 30 2-fold axis of symmetry. So if you can see here, there are different types of viruses, different sizes, which all have these axes of symmetry. And typically the axis of symmetry is formed by protein molecules, which make up the outer shell of the virus. So the protein molecules have to come together in a very specific manner in order to generate the shell, which keeps up or which agrees with the principles of icosahedral particle formation. So the formation or the assembly of the particles is dictated by something called a triangulation number. And I won't go into details, but these are smaller particles with lower triangulation number, and then you can see particles which have higher triangulation numbers like 7 or 25, etc. So the question that we were very interested in knowing is, how does disassembly work? When a virus particle is falling apart, does it just burst like a balloon, or does it become gradually asymmetric? So instead of being a very sort of symmetric particle, does it gradually start perturbations in various parts, or do the perturbations start from any particular part of the virus? So that eventually it falls apart and the genome that was inside can be released for downstream replication and transmission. We were working on a model virus called flock-hood virus, and this is what the virus typically looks like when we do negative things. Now we can mimic disassembly conditions in vitro. So instead of being inside a cell, we have purified the virus, and the virus is in tube. Now, as we gradually heat the virus, we start seeing that the virus is changing its conformation, its structure. And eventually we reach a point where we see a puff of RNA. This is an RNA virus. So a puff of RNA is coming out of the virus. So this observation by electron microscopy immediately told us that the entire particle does not seem to be bursting like a balloon. Rather, the RNA seems to be associated with one specific edge of the virus, indicating that there could be some kind of a weak link in an icosahedral, very symmetric icosahedral particle, which may be exploited for release of genome. So we used, now here I should mention that this population contains a variety of particles. This is a heterogeneous population because all the wild type particles have not been converted to different disassembly intermediates. So to speak, maybe the majority of the particles are in a specific step of disassembly, but some particles could still be in native state and some particles could be in an advanced state of disassembly. So this is a sample that we cannot actually resolve using either X-ray crystallography, and NMR is out of the question because these are very large particles. This is a virus particle, almost 9 megadalton, so this cannot be resolved using NMR. So we utilize cryo-electron microscopy and 3D reconstruction to look at the particles. So here, what we did was, if you remember the 2D classification method, we individually boxed these particles, and then we did a 2D classification, which showed us that some particles may have been of native type, some particles may have been of different types of intermediates. So this is the wild type native particle on the left-hand side. And on the right-hand side is the first stage or the first intermediate that we were able to arrest in the disassembly field. So if you see, this is what the wild type particle looks like, and this is what the first intermediate looks like. And it's very clear that the first intermediate has significant bit of density missing or disordered, right in this region. So there's a significant amount of density which is disordered or missing. And when we looked at it in more detail that I will not go into, we found that the disorder starts from one of the two-fold axis of symmetry of the virus. So it appears that the two-fold axis of symmetry perhaps is a little less robust than the three-fold or the five-fold, or there could be other reasons why the two-fold undergoes disorder at a little more quickly than the other axis of symmetry of the virus. Now, we were also able to look at another intermediate. Now, this intermediate is the one where you see the virus particle here, and it's associated with the puff of genome. So the RNA is coming out, and these are called puffed particles. So these particles were mixed in with intermediate one, and this is where the beauty of electron microscopy comes in, that you can individually look at these particles and separate them out from the first intermediate. And we first looked at just the portal region. So we looked at the region right here, right in this region, where the genome first starts to get released from the virus. So we can see that there is connectivity of this density from the puff to the interior of the virus particle. And you can see this very clearly in the central section, that the puff of density is connected to the interior RNA of the virus. And this tells us that the way, and once we resolve this in high resolution, it will tell us, I hope, in more detail, how exactly the RNA comes out, what are the regions of the capsid proteins, very specifically, which alter their conformation to allow the RNA molecule to come out, what are the interactions between RNA and protein, which are disrupted during this release of RNA from the viral genome. So we're hoping to get all of this information. We already have some information. We're trying to get very detailed information. So we can tell, using cryo-electron microscopy and 3D reconstruction, how a virus particle goes from here to here, where it is partially disordered, and finally to here, where the genome is coming out. And I'm sure there are other stages in between here and here, which can be resolved using cryo-electron microscopy and 3D reconstruction. So in sum, we have looked at how electron microscopy has evolved over the years, and what is cryo-electron microscopy and its impact on biological macromolecules. why is this important? How does this help advancement in our knowledge? How does this help us to generate drugs or therapeutics? And how does it help us to understand fundamental biological processes, which could be very difficult to follow using either biochemical methods or standard or typical, or I shouldn't say standard because cryo-electron microscopy and 3D reconstruction is a very standard method now, but relatively older 3D structure resolving methods like X-ray crystallography or NMR. So with this, I hope that I have had some of you interested in biological electron microscopy, cryo-electron microscopy, 3D reconstruction, and structural biology. And I will end here. Thank you very, very much for your attention. And I'm happy to address questions.
Thank you, ma'am. Thank you very much for the informative talk. Now, there are a few questions that I have posted in the chat box, Meet chat box. If you can kindly take them up.
Yes, let me go there. Yes. Shall I stop presenting? Would that be helpful?
Yes, ma'am.
Okay. So first question is from Rajesh Khanna, who wants to know, shall we analyze protein receptor interactions using cryo-electron microscopy? Yes, absolutely. This is being done for a variety of protein ligand, protein receptor, virus receptor. All of these has been utilized. It's a good way. Cryo-electron microscopy is a good way to analyze protein receptor interactions. The next question again is from Rajesh Khanna, who wants to know, with structural information, shall we design inhibitors? Absolutely, this is what people are doing. In order to design inhibitors, you have to get very high resolution information. So if your resolution is stuck at 4 angstrom or 3.5 angstrom, then it's very hard to design, or try to figure out how a small molecule binds to specific amino acids in a protein molecule. But recently cryo-EM has broken the resolution barrier, so routinely people are generating structures which are 2 angstrom resolution, less than 2 angstrom resolution. So it is absolutely possible to design inhibitors. Now, the third question, also from Rajesh Khanna, is what is the resolution difference between X-ray diffraction and cryo-electron microscopy in protein structural information? Now, this is a very interesting question because there is so much controversy about it. And if you are interested, you can email me and I can, send over a lot of published articles which discuss, how to calculate resolution in cryo-electron microscopy. So, since the two methods are the calculation of resolution is not using the same method, because resolution is calculated using different methods in X-ray crystallography and cryo-electron microscopy, I wouldn't say that you can compare them side by side, but cryo-electron microscopy can at this point resolve protein structures at a resolution of less than 1 angstrom. So that's a tremendous advancement, and it is routinely it is possible to see amino acid side chains in the method. So it is very applicable. It is absolutely useful for designing inhibitors, and that is what a lot of people are doing for these SARS coronavirus 2. The fourth question is from Pragya Bradu, who wants to know, since bacteria and viruses are studied using the cryo-electron microscope, what is the process of cleaning the microscope after use, along with all the safety measures? So when you freeze your sample, so firstly, in a facility where you're using a cryo-electron microscope, in addition to the microscope, you have a lot of other equipment. You have equipment to treat the grids. You have equipment for freezing your sample. You have to bring your sample in. So typically for bacteria and viruses, there are various safety levels, right? So there is biosafety level 4 is the highest safety level. Then you have biosafety level 3. Your current coronavirus can be studied in biosafety level 3. Then you have biosafety level 2 and biosafety level 1. So usually, let's say for very highly infectious particles or bacteria, viruses, etc., you have to have your microscope in an appropriate biosafety level facility. Because it's not only the microscope, it's your sample and it's all of your sample treatment, equipment. But having said that, after you've frozen your sample, it is no longer moving around. So when you put it in the microscope, the idea is that it should not disseminate in the microscope and contaminate the microscope from within. However, chances of contamination are high when you are actually freezing the sample, or the point where you are actually, manipulating your sample, taking it out of your tube, putting it on the, grid, freezing it, putting it in the microscope. So during those processes, you want to be in an appropriate biosafety level facility. It is very, very important. The last question, I think, is, shall we determine the charge of protein? I'm not sure what this means. I'm going to try to address this in a general way. So usually what happens is, your protein molecules can be hydrophilic or hydrophobic. So usually when you have your grids, you're trying to put your sample on grid, you try to give some sort of negative charge to the grid so that your sample can bind. On the other hand, if your sample is primarily hydrophobic, then getting it into the holes becomes a challenge. And that is when one uses different types of grids, like gold grids or carbon coated grids, which are little more hydrophobic in nature, so that your sample can associate or bind a little bit better. So that then becomes important. Then I think Basu Bhattacharya wants to know, can the cryo-EM images be free of the charging effects we usually see in FESEM or FETEM analysis of biological samples? So I'm not sure about this because I don't know what charging effects you see in FESEM. But typically, there should not be any charging effects in cryo-EM images. These are just flat 2D images that you're collecting. And then you have to try to generate a 3D structure from the flat images.
Thank you, ma'am. Thank you for addressing all the questions. Now I would request Dr. Rajiv sir to kindly give the vote of thanks to the speaker.
Good evening, Professor Banerjee. So I thank you for accepting our invitation and delivering such an amazing topic. And as I was following the views of the presenter in YouTube, where your broadcast is, this was broadcasted live, so I could find that it is really an amazing feedback that we have received for these deliberations. Thank you, ma'am, that so many participants in our country got enlightened with the recent advances of this particular technology and the science behind it. So I, on behalf of NIT Silchar, express my gratitude, and I express my heartfelt thanks from the organizer side for sparing your time and sharing your expertise with a wider community. So thank you very much once again.
Thank you very, very much for inviting me to present. And I am very, very glad that, if anybody got interested in the topic. And thank you. Thank you very much.
Thank you.
Thank you, ma'am. Thank you very much.
So, dear participants, thank you very much for patient hearing and participating in an hectic or you can say marathon session that we are having. So thank you for your patient hearing and submitting the feedback also from time to time very seriously. But we have one request from the organizer side is that while the lecture is going on, please don't keep posting about the feedback. Doesn't look nice. So we will be posting the feedback at the end of the lecture. We have already announced that. And so please be patient. And while the deliberation is finished, at that point of time, we'll give you the link. Now the link is open for this session. We'll keep it open for some time now. So please give your feedback and be patient. That will be a request from our from organizer side. So we will sign off now, but the feedback will be open. You keep sending your feedback for another 5-10 minutes. We'll keep it open and see you tomorrow morning again. So we have a couple of nice lectures tomorrow, okay, from different perspectives. So I hope you will enjoy them. And till then, see you. So we are signing off now from here. And stay safe and stay healthy. Goodbye. Good night.